Today I'll discuss the citric acid cycle, Krebs cycle, or the TCA cycle. So as we know, before entering the TCA cycle, the pyruvic acid is formed in the glycolysis. So the pyruvic acid thus formed enters the link reaction in the cytosol itself, where it is converted into the acetyl coenzyme A. After that, the acetyl coenzyme A will enter the mitochondrial matrix and start the TCA cycle. So let's see. The acetyl coenzyme A will combine with a 4-carbon compound which is known as oxaloacetic acid. This is a condensation reaction by the enzyme citrate synthetase to form a 6-carbon compound called as citric acid. After that, the citric acid will convert into cis-aconitic acid by the enzyme aconitase, in which Fe2 plus iron is used as the cofactor. This is a dehydration reaction, so water is eliminated in the process. Next, the cis-aconitic acid so formed will convert into isocitric acid. Again, the same enzyme is used, that is aconitase, and Fe2 plus is used as the cofactor. But this time it is a hydration reaction, that means water will be added in the process. Now, the isocitric acid will convert into oxalosuccinic acid. This is an oxidation reaction by the enzyme isocitrate dehydrogenase. So, one NAD molecule will be converted into NADH molecule. NAD converting into NADH molecule. Now, oxalosuccinic acid will convert into alpha-ketoglutaric acid. Now, this is a decarboxylation reaction by the enzyme oxalosuccinate decarboxylase. So, one CO2 molecule will be eliminated. So, now oxalosuccinic acid converted into alpha-ketoglutaric acid, which is a 5-carbon compound. Why? Because one CO2 molecule is eliminated in the process. Alpha-ketoglutaric acid will again convert into succinyl coenzyme A, which is a 4-carbon compound. And the enzyme used is alpha-ketoglutarate dehydrogenase. This is an oxidative decarboxylation reaction. Why? Because alpha-ketoglutarate acid oxidizes into succinyl coenzyme A, and in the process, one CO2 molecule is eliminated. So, again, an NAD molecule is converted into NADH, and one CO2 molecule is also eliminated. In this process, there are certain cofactors that are used, that is TPP, thiamine pyrophosphate, H is coenzyme A, lipoic acid, and magnesium ions. Succinyl coenzyme A will convert into succinic acid, which is again a 4-carbon compound, by the enzyme succinate thiokinase. Now, this is a substrate level phosphorylation reaction in which the ATP is generated. So, the ADP plus inorganic phosphate will form an ATP molecule. Succinic acid is formed. Now, succinic acid will convert into fumaric acid. Now, again, this is an oxidation reaction, so by the enzyme succinate dehydrogenase, but this time an FAD molecule is converted into FADH2. An FAD molecule is converted into FADH2. Now, fumaric acid thus formed will convert into malic acid by the enzyme fumarase. Now, fumarase is a lyase family enzyme in which water is used to break the bonds. So, malic acid is formed. Now, malic acid, a 4-carbon compound, will convert into oxaloacetic acid, which is the first step. So, there is a regeneration of oxaloacetic acid by the enzyme mannate dehydrogenase. So, again, there is a formation of NADH molecule. So this looks a bit complicated, but once you get it, you will remember it forever. You will have to remember it for the energy dynamics, which we will see soon. Now let's see the energy dynamics of the entire cycle. From glycolysis, we know that one glucose molecule is broken down into 2 pyruvic acid, and in the process, 2 ATP molecule and 2 NADH molecules are released. So 2 ATP and NADH, we know 1 NADH is worth 2.5 ATP. So 2 NADH molecule will be worth 5 ATP. So if we do in total, there are 7 ATP molecules that are released from the glycolytic pathway. Now from the link reaction, the two pyruvic acid molecules are broken down into two acetyl coenzyme molecules and two NADH molecules are released in the process. Again, one NADH is worth 2.5 ATP. So two NADH molecules will be worth 5 ATP. So here we get 5 ATP. So from the link reaction we are getting 5 ATP, from the glycolysis we are getting 7 ATP. In the TCA cycle which we have discussed in the lecture, we started with one acetyl coenzyme molecule, but we know that the 2-pyruvic acid that we get from the glycolysis that breaks down into two acetyl coenzyme molecule. So whatever we get here as a product will be multiplied by 2. So if we get 3 NADH molecule, that will be multiplied by 2, that is 6 NADH molecule. So here we get 6 NADH molecule and 2 FADH molecule, and this is 180. If we calculate this, we will get 10 ATP. So I'm repeating again, we get 7 ATP from glycolysis, 5 ATP from the link reaction, and 10 ATP from the TCA cycle. If we add 3 of them, that the entire cycle, we get 32 ATP in total. This was the entire TCA cycle and the energy dynamics. Thanks for watching.